Today I'm going to talk about preterm birth and how it relates to the fetus's support structures, the placenta and the fetal membranes. As you can see from this cartoon, these structures are genetically related to the fetus. Worldwide, a full 10% of babies are born preterm or prior to 37 weeks of gestation. That leads to a total of 15 million babies born too early every year around the world. This same statistic of 1 in 10 is the preterm birth rate here in the United States. In the US, that 10% prematurity rate becomes a bit more complicated when you break it down according to the race or ethnicity of the mother. And there are local variations also from state to state within the US. However, overall, the average of 1 in 10 is fairly robust. There are two things we can take from this discrepancy within race in the US. One is that there is probably a lot we can do to improve the overall preterm birth rate by eliminating racial disparities. That could be done with a combination of social, economic, and care factors. But two, there will still be a relatively high proportion of babies born preterm, even if we address these racial discrepancies. And for our ability to address that, we require basic physiological understanding of why preterm birth happens in the first place. Preemies' outcomes are worse the earlier they are born. So a close to 37-week gestation is not associated with that much different mortality than a full-term baby. But as you can see, moving towards the left on the x-axis here, going to earlier and earlier times of gestation at the time of birth, you see that the mortality rate starts to increase, and at 27 weeks, which is the boundary between the second and third trimesters, the mortality rate is about 20%. In addition to greater infant mortality for preterm birth before 27 weeks of gestation, there are also far greater risks of complications, particularly those to do with the lungs and the respiratory system, which are very close to 100% at that boundary of 27 weeks between the second and third trimesters. Compounding all of this is the fact that our 1 in 10 number of preterm births is stubbornly constant. Improvements in technology are not significantly changing the baseline preterm birth rate. Therefore, a new approach is needed. Here's where we as bioengineering and biomaterial scientists could perhaps play a role. In order to address diagnosis, prevention, and treatment for preterm birth, we first need better understanding of the basic physiology of why and how preterm birth happens. Let's start at the very beginning with a look at the fertilized egg and its first steps of differentiation, even prior to implantation into the wall of the mother's uterus. The fertilized egg differentiates into two cell types straight away: the inner cell mass, which will become the fetus, and the trophoblast, which is the peripheral cell layer that surrounds the inner cell mass. That trophoblast is what will directly contact the maternal decidua and then invade into that decidua as the implantation takes place. This interaction between the maternal decidua and the fetal trophoblast is the direct interface between two genetically distinct individuals. The trophoblast will go on to form the placenta, and that will be the crucial immune interface between the mother and fetus. This early differentiation of the trophoblast layer is the start of the formation of the extraembryonic fetal tissues, the placenta and the fetal membranes, that surround the fetus and support the fetus in terms of providing oxygen, nutrients, and an immune interface with the mother throughout gestation. To allow for studying this complicated biological system in the laboratory, we will use hydrogel-based biomaterials with a range of different compositions and different cross-linking mechanisms at the molecular level. Both chemically and physically cross-linked hydrogels have played roles in the in vitro investigations of these early processes of pregnancy. We will begin here by looking at processes that take place in the first trimester of development, the trophoblast invasion and the development of the placenta, that have third trimester implications in clinical conditions such as fetal growth restriction in 3 to 10% of all pregnancies and preeclampsia in 5 to 14% of all pregnancies. These are very large problems that develop clinically in the third trimester, but which have their roots in the first trimester in insufficient trophoblast invasion and poor early development of the placenta. The picture of trophoblast invasion here illustrates that in some ways this is a Goldilocks process. We need the trophoblast cells to invade into the maternal decidua, or the lining of the uterus, just far enough, not too far and not insufficiently far. This process of trophoblast invasion is related to the early development of the placenta in that the trophoblasts actually help to remodel the maternal spiral arteries and form this early interface between the mother and fetus. In a preeclamptic or fetal growth restriction case, the cells do not invade deep enough into the maternal decidua, and it's unknown why. It's unknown what types of cues are causing the trophoblasts to know how far to invade, whether they're biomechanical, biochemical, biological, or some other combination of all of the above. In order to study this process of trophoblast invasion, we've developed a microfluidics assay which takes trophoblast cells from the first trimester, puts them in a hydrogel channel inside a microfluidic device, and then passes two different liquids of different composition on either side of the hydrogel, allowing us to establish a chemical gradient across the gel due to the presence or absence of a biochemical factor in one liquid relative to the other. The pictures on the left illustrate the formation of the chemical gradient in the hydrogel in that central channel as a function of time, and this is also plotted on the graph here. As you can see, the width total of the channel is about 1.3 millimeters, and as you introduce, in this case, a fluorophore, just for visualization purposes, you can see that the diffusion progresses across the hydrogel, and within 12 hours, a stable gradient is set up across the hydrogel in that central channel of the device. We can then take a confocal microscope and take image stacks at different locations throughout this microfluidic device and use that to track the individual cells' movements as a function of this chemical gradient. And we can do so by measuring two different parameters here illustrated on the bottom of the slide. First, on the left, we can look at directionality, which is essentially how directed the motion is towards one channel or another, defined as the length of the distance between the start and finish points of the cell divided by the more tortuous actual path. We can also look at the motility, which is the total number of cells moving towards either the A or B channel divided by the total number of cells. These plots show the individual tracks of a subset of cells. On the control, on the left-hand side, there is no chemical gradient, whereas on the right-hand side, there is a chemical gradient with a molecule called GM-CSF placed in one of the two channels. As you can see on the left in the control, in the absence of a gradient, the cells do not move in a directed manner, whereas in the presence of a gradient, the cells move in a very directed manner. When we quantify this in terms of our directionality and our motility, as you can see, the directionality is slightly increased with the GM-CSF chemical gradient. The motility, or movement towards channel A, is substantially increased, with almost all of the cells moving towards the chemical of interest. On the upper left-hand corner, as you can see, there's not a big difference between the velocity of the cells. So even though they're moving in a more purposeful manner towards the chemical gradient, they are not moving any faster on average. Now, as I said, we put these cells in a hydrogel, in Matrigel, in this device, but this is a question in terms of how well this Matrigel actually mimics the maternal decidua. And in order to investigate this question more, we have to define some different regions of the maternal decidua relative to where the fertilized egg has actually implanted. So the decidua basalis is the area where the implantation has occurred and where the placental development starts to happen. The decidua capsularis is essentially the covering of decidua over the implanted fetus, and the decidua parietalis is the distant region away from the implantation site. We will not focus any more time on the capsularis here, but we will compare the basalis, or implantation site, with the parietalis, or distant from the implantation site. What we've done here is looked at the mechanical properties as measured with an atomic force microscope indentation test for comparing in a series of individual patients the decidua basalis and the decidua parietalis. And as you can see, both in the graph on the left for the individual patients and then the graph on the right for the combined data, there are significantly stiffer regions of the decidua basalis at the implantation site compared to the parietalis, and the parietalis is actually comparable to both the non-pregnant endometrium and to the placental tissue at third trimester. This is a graph showing those results in summary, and the black curve there is for Matrigel, the hydrogel that we were using in the microfluidic device. And as you can see, depending on the concentration of the Matrigel, going from 40% to 100% as reconstituted according to the manufacturer, the stiffness of the Matrigel is reasonably comparable to the non-pregnant endometrium, to the placenta, or to the decidua parietalis distant from the implantation site, but significantly less stiff than the decidua basalis where the implantation site has occurred. So in fact, this may not be the best case scenario in terms of understanding these interactions between the trophoblast cells and this artificial extracellular matrix, and this calls for some potential thinking about development of a new hydrogel for this microfluidics assay. We are not the first people to think this, and there have in fact been recently quite a few papers published on synthetic alternatives to Matrigel. Now we can look at what Matrigel is on the left. It is a naturally derived hydrogel, and important from the perspective of the placental and decidual tissue is that it's very high in collagen type 4. Collagen type 4 is really interesting because it is present in the basement membrane of mature tissues all throughout the body, but it seems to play a unique role in pregnancy and is present in unusual ways in three-dimensional structures in pregnancy. The picture on the right is an example of a synthetic scaffold that is being used to study different types of cell differentiation and to address some of the issues that arise with Matrigel. It has significant batch variability, and that leads to variations in both its composition and also its mechanical properties, which from our view as bioengineers is a particular problem. However, one thing that the design of these types of scaffolds have to take into consideration is the mechanism of bonding within the hydrogel. So if we take here, we have a plot of elastic modulus versus the intrinsic permeability. The permeability is essentially a measure of the pore size within the hydrogel, and it relates to how easy it is for fluid to move through the hydrogel or artificial extracellular matrix. These are measurements we've made with indentation testing. And what you can see is that there's a big difference between physically cross-linked and chemically cross-linked gels in this relationship between the elastic modulus and the permeability. Both of those parameters are fundamentally related to the microstructure of the gel and the pore size, but as you can see, the chemical gels have some relationship to some mature tissues here, and we don't actually know where the decidua fits on this curve because we haven't yet measured its permeability. And so this is part of the ongoing work, thinking about what's in the natural decidua, in our case particularly being interested in collagen type 4, and thinking about how to make a Matrigel replacement that is both rich in collagen type 4, but also that replaces the material that is so variable and so dependent on the manufacturer with something that's very consistent, and not only consistent in composition, but as a result consistent in terms of these mechanical properties, both the elastic modulus and the permeability. So far, we've been talking primarily about the very earliest stages of pregnancy in the first trimester, from the implantation of the fertilized egg into the maternal decidua, after this first cell differentiation into trophoblast and inner cell mass, and then in the subsequent weeks as we start to see the early development of the placenta. But now I'd like to turn attention to the much later development, so in terms of months of development of the fetus, and look at a second and third trimester problem, which is premature rupture of the fetal membranes. So the preterm, pre-labor rupture of membranes, or PPROM, is actually responsible for a full third of all preterm births. And that means that it's present in about 3% of all pregnancies. This is a very familiar trope to most people, because if you've ever watched a movie in which there's a pregnant woman, at some point she's probably in an elevator or otherwise inconveniently located when the membranes rupture and her shoes are wet. Now, this is as I said, a very cinematic trope, but at the same time, this is a very serious problem. First of all, because most of the time preterm membrane rupture does not have a known cause. And second of all, as you can see here, oftentimes it occurs in the second trimester. And at this point, there's no way to fix that membrane and carry on with the gestation, and the risk of infection becomes great, such that the baby is usually born by induction or C-section in order to prevent infection. It is interesting to note that the mechanics of birth have been studied from a rigorous and scientifically sensible perspective since all the way back in the 19th century. This is a paper that was published by a medical doctor in a reputable journal in 1867, considering the forces associated with the uterine contraction and natural labor, thinking about this question of how much force is associated with the birth process, and therefore how much force is associated with the rupture of the membranes. In order to look at this fetal membrane rupture in more detail, we need to look at the microstructure of these tissues. So if we take a blow-up from the right-hand side moving towards the left-hand side, and we look at the fetal membranes, there are two layers, the amnion and the chorion, and it is the internal or baby-facing layer of the amnion that is most important biomechanically. This is because the tissue is primarily made up of fibrillar type 1 collagen, and it has an epithelial cell layer on the intersurface that helps to maintain the amniotic fluid pressure and volume as the gestation continues. So continuing to zoom in on this microstructure, I show the schematic here along with both a scanning electron microscope image on the right as well as a histological image. And what you can see is that this really is a tissue that is primarily made of collagen. There are not very many cells here at all, and what cells there are mostly on that epithelial surface. There are a few cells scattered within the fibroblast layer, but the compact layer, which is thought to be most important mechanically, is really almost entirely collagen. Now, because we want to study the process of fracture of this membrane at the time of birth, we have been conducting a series of tests where we've been studying tear and fracture toughness using a couple of different geometries. So we've been using a trouser tear test, which is exactly as it sounds and as it's depicted here. It's like how you would tear a piece of paper by pulling your hands in opposite directions. And we've been comparing this to a more mechanically rigorous pure shear fracture toughness test in which you grip a small section of tissue relatively close in the grips and then introduce a notch and propagate that notch. The analyses for both of these types of tests were actually considered quite a bit in Rivlin and Thomas's 1953 masterwork on the fracture of rubber. And so the analyses for these are pretty straightforward in that they have been considered before. And in the case of the pure shear specimens, we use a unnotched control specimen along with the notched specimen to calculate the fracture resistance. In the case of the trouser tear, there's a simplified analysis that works quite well. These are pictures of the amnion in the process of cracking. So these are cases where we've introduced a notch with a scalpel, as you can see in the pictures, and then propagated the notch. As you can see, there's a significant difference between the smoothness of the scalpel cuts and the roughness of the ruptures as the crack propagates through this collagen network. And interestingly, when we look at the raw data for our specimens, there's actually not a significant difference between the stress-strain response as we pull on these specimens, regardless of whether they have a notch cut in them or not. So the mechanical response is relatively notch insensitive, which is an indication of just how tough the tissue is. When we look at the quantitative fracture results, we see that we get a pretty substantial fracture resistance value, or 1 to 2 kilojoules per meter squared. And this is pretty comparable for some other soft tissues, as we'll see in a moment. But this is now our target. So we now know what this natural tissue has in terms of its fracture properties, and we can think about what kind of material we can design that might be suitable for a patch for the membranes or as just a study system for examining these membrane behaviors in the lab. So what we've been doing is using electrospinning to create a fibrillar network that looks very much like this dense type 1 collagen network that's present in the native tissue. So we use gelatin in an acetic acid solution, so something that's not terribly toxic and not a horrible solvent. We use a 15 kilovolt voltage generator and a static target to make randomly oriented gelatin fibers, which are quite comparable to the collagen fibrils. And then we infiltrate these fiber mats with a hydrogel. And in this particular case, we're using alginate, which is a seaweed-derived sugar, and it's physically cross-linked with calcium to cross-link. We've also played with chemically cross-linking the gelatin itself, but I won't have time to go into those results today. When we measure the fracture toughness of these materials, we can get quite a range of behaviors depending on how we orient the fibers within the gel. So if you look on the very left-hand side, we have the toughness of the base material, so the gelatin gels and the alginate gels, and it's very low. Then as we add the randomly oriented fibers, we get an increase in the toughness by about an order of magnitude to 0.2. Then as we orient the fibers a little bit better and make sure that we have fibers present in different orientations, we can see further increases in the toughness of these composites that we've made in the lab. And importantly, if we compare these composites that we've made, they're much closer to the properties of the native soft collagenous tissues, including amnion, which is shown on there with a couple different green stars with the letter A in it, reflecting a couple of different studies. We've gotten slightly different values of the strength and toughness on average in the past couple of studies that we've done. But in either event, what's really important about this plot is that the single network hydrogels have nowhere near the mechanical properties of this natural fibrillar amnion tissue, whereas the composites that we make with fibrils in them do start to have mechanical properties that are comparable in both strength and toughness to the native soft collagenous tissue, including amnion. So today, in summary, I have talked about two different approaches in which we've used hydrogels for very different purposes in the process of studying pregnancy and reproduction. So first of all, we looked at hydrogels for trophoblast invasion, considering the fact that we need to control the pore size and the permeability of the hydrogel in order to establish our chemical gradient, and that permeability and the stiffness probably both contribute to the control of cell migration. And overall, this study was extremely promising from the view that we can see very clearly that we have directed trophoblast migration in response to a chemical signal, but we need to do more work to tailor the decidual-like hydrogels for our future studies. And second, we used hydrogels as a matrix in making a biomimetic hydrogel nanofiber composite material in order to mimic native amnion. And we used this system to try to tune the fracture properties to match the properties of natural amnion, and consider this either as some sort of an in vivo patch or as a scaffold for doing ex vivo research on these fetal membranes, including some more biology. So with that, I hope I've given you a crash course in the fascinating world of the extraembryonic fetal support structures, the placenta and fetal membranes, and how we can use hydrogel biomaterials to study and mimic these structures, and hopefully to try and gain greater insight into the processes that lead to preterm birth in pregnancy. Thank you. And with that, I'd be happy to take any questions.